So thanks, John. I really appreciate this opportunity to give a brief overview of some of our more recent research on these electroactive microbes that can exchange electrons with extracellular minerals or other microbial species. And you mentioned applications. If I have some time, I'm primarily going to talk about the geomicrobiology, but I will talk about, if there's time, some of these spin-offs of novel electronic materials that we can produce with either the electroactive microbes or at least their genes, and have been used now and recently in several novel electronic devices. But I do want to focus on more of the biogeochemistry. And just so we're all starting at the same place, I'd like to point out that, of course, electroactive microorganisms have important implications in a wide diversity of environments, possibly even in our gut system, and that there's a wide diversity of microorganisms that are electroactive. And this is not only in the bacteria, but also archaea. All right. So I'm primarily going to focus, sorry, I'm just trying to move this thing out of the way. Guess I can't. I'm going to primarily focus on anaerobic soils and sediments in the subsurface, and give some examples from each of these major redox zones. So first I'll talk about some new research on electroactive microbes in iron-reducing environments, and then something about sulfate reducers, and then methanogenic environments as well. So looking at the iron-reducing zone first, today I'd like to focus on what are called the respiratory electroactive bacteria that can completely oxidize acetate and other fermentation products to carbon dioxide with the reduction of iron. And the reason for focusing on those is that they account for most of the iron reduction in most soils and sediments. And the first microbe found to be able to carry out that reaction of completely oxidizing organic compounds to carbon dioxide with iron as the electron acceptor was Geobacter metalloreducens. It can grow on an insoluble iron oxide that's produced in the laboratory. And here's a scanning electron micrograph, which shows Geobacter colorized in green. It's growing on this insoluble iron oxide, oxidizing, in this case, I believe, acetate to carbon dioxide with the reduction of iron. So a lot of what I'm going to talk about for iron reduction will focus on Geobacter. I know other organisms like Shionella are more intensively studied in the laboratory, but there's nothing like Geobacter for microorganisms that can completely oxidize organic compounds to carbon dioxide, which Shionella can't do, and also for rapid growth on iron oxide. Furthermore, when we and others look at the microbial community in soils where iron reduction is taking place, and you can often see this reaction as the reddish-brown iron oxides are stripped out of the soil, giving this more gray, glade soil. These molecular analyses typically reveal that Geobacter and closely related species are the most abundant and metabolically active microbes in a diversity of soils, sediments, and subsurface environments where iron reduction is important. And we see this predominance of Geobacter not only in those pristine environments, but also when subsurface environments are contaminated with organics. Petroleum is a typical groundwater contaminant. There's often a bloom of Geobacter with iron reduction taking place. Again, you can see here's a pristine soil, up gradient of a jet fuel spill. Here's down gradient. You can see the soil is gray. It's due to the abundance of Geobacter species, which metalloreducens was the first organism of any kind, not just an iron reducer of any kind, though, that could anaerobically oxidize these aromatic hydrocarbons, including benzene, which at the time always was thought it would persist under anaerobic conditions. And Geobacter and other iron-reducing microorganisms can substitute other radioactive and toxic metals for the iron in their respiration. This was also first seen with metalloreducens. And this is an important reaction biogeochemically, because for uranium, soluble uranium-6 is reduced to highly insoluble uranium-4, often forming uranium minerals such as uraninite. And this can be a bioremediation strategy when the subsurface is contaminated with uranium. You just pump some fluid like acetate into the subsurface to promote the growth of the Geobacters. So we're really lucky that because Geobacters are the important organisms in many of these environments, that they're actually we were able to recover them in culture, and they're genetically tractable. So this has made it possible to have a more systems biology approach to studying some of these subsurface processes. But that's been really limited to understanding the central metabolism and modeling that, because what's been lacking in these models is a real good understanding for the real, I think the important question for most electroactive microorganisms is how do they get the electrons that are generated from inside the cell, from central metabolism, outside the cell, onto these insoluble electron acceptors, such as iron oxide in this case. And many of these electroactive microorganisms, if they're gram-negative, achieve this through what's known as a porin-cytochrome system. I'm going to talk about this quite a bit today. It's 3 proteins, a periplasmic facing cytochrome, a beta barrel porin protein, and an outer surface facing multi-heme C-type cytochrome. In addition, they often have other multi-heme C-type cytochromes, so I'll show you in Geobacter, that can give you more electrical contact. But the question has really been, many microorganisms have this kind of setup. I mean, Schumann was the best studied example, but many have these cytochrome porin systems, and yet Geobacter just reduces iron so much faster and seems to predominate in many of these environments. And the question has been why. So I'll give you a little background first. Sorry for all the historical background, but it's, I think, necessary to explain the new results. And the reason why dates back to this observation made by Susan Childers almost 2 decades ago now, that when Geobacter metalloreducens is grown on insoluble electron acceptors like manganese or iron oxides, it expresses pili and flagella that it doesn't express during growth on soluble electron acceptors. And it turns out that the pili really have a key role in the iron oxide reduction. If you delete the genes for these pili, the capacity for iron oxide reduction is removed. And as seen in this transmission electron micrograph, iron oxides tend to specifically associate with those pili, and it turns out that they're electrically conductive. And their conductivity is more than sufficient to account for long-range electron transport to iron oxide. That was determined through studies of the conductivity of individual pili. And that's for Geobacter. Sulfur-reducens for metalloreducens, which has more aromatic amino acids, and aromatic amino acids confer the conductivity. The conductivity is even higher. This is, I think, the most conductive biological protein wire that's known. You can also change the conductivity of these wires by pulling out some of those aromatic amino acids. So I'm going to talk quite a bit today using this strain ARO5, where we took some of the aromatic amino acids out of the pilin monomer that assembles into the conductive filament. And as you can see here in these measurements of individual conductivity, that here's the wild type pilin, here's the ARO5 pilin. It has extremely low conductivity, 1,000-fold less than the wild type. And that's important then when you want to study long-range electron transport in Geobacter strains, to have this kind of strain, because you can still express the pili. So the other pilin functions that may be involved, say in attachment or motility, are retained, but the conductivity is lost. And you can see here, when that conductivity is lost, so is the capacity for iron oxide reduction. So this is our working model up until very recently for long-range electron transport. Electrons released from organic matter inside the cell, move out along the electrically conductive pili onto the ferrochromium. So we're basically done with that model, and we're moving on to other things. When a couple of papers came out within the last couple of years in fairly high impact journals that said that this model was completely wrong. And the reasoning for that was that when you look at preparations of proteins harvested off the outer surface of the cells with cryo-electron microscopy, it's difficult to see these pilin-based filaments. So we were really surprised that our model was thought to be wrong, especially since even before that, there was just so much data that would refute the model based solely on cryo-EM observations. Especially, you have to remember, to do cryo-EM, there's many purification steps and preparation steps that may cause artifacts in the analysis. In other words, I'm not going to go through all these results because I'm going to show you the new results that further confirm the presence of the pili and their function in long-range electron transport. So as I say, we were thought we were done with this, but I think it's great to have challenges and what are often accepted ideas. And so we went back to look at this question again with a totally new method, which is direct observation of untreated cells with atomic force microscopy. And the reason we like this method is it avoids any processing at all. So in this method, we just take cells of Geobacter, in this case, it's a sulfur-reduced hand strain, drop cast them on a flat conductive substrate, and just look by atomic force microscopy to see what we can see. And what we see here are multiple cells of Geobacter, and of course, many filaments emanating from the cells. If we zoom in with this white, where this white box is here, we can take a look and see what kind of filaments are emanating from the cells. So the alternative hypothesis for the electrically conductive pili concept is that all the long-range electron conduction goes through a multi-heme C-type cytochrome filaments. And certainly those filaments exist, and we knew that for quite some time. And here's an example of one of those cytochrome filaments. It's comprised of the multi-heme C-type cytochrome, OMCS. I've designated it here with the red arrow. How do we know that's OMCS? We can tell by its diameter. It's 4 nanometers, and a very characteristic longitudinal pitch, repeating at 20 nanometers. So that's clearly OMCS. That's the OMCS filament structure. However, if we look at the totality of filaments emanating from the cells, we see that 90% of the cells, as indicated by the blue arrows, are not OMCS. Instead, they're filaments of 3 nanometer diameter. So this is exactly the diameter that had always been reported for the pilin-based filaments. Yes, and when we look at the conductance, this is now using a conductive tip, and just looking at conductance across the pili, we see that they're just about the same. The OMCS conductance slightly less than that of the 3 nanometer filaments, which we believe are e-Pili. To look at further whether these were e-Pili, we took that Arrow 5 strain I described before. So this again is the strain expressing the poorly conductive filaments. When we look at this strain, what do we see? The same distribution of filaments as we would expect in the wild type. 90% or a little bit more are those 3 nanometer diameter filaments, but there's a drastic change. Although the OMCS filaments still have their same conductance, you would expect that. You shouldn't affect the structure or conductance of a cytochrome filament by changing the structure or the amino acid content of the pilin. But note that the 3 nanometer wire now has a conductance that's orders of magnitude lower than what it was in the wild type. So clearly these 3 nanometer filaments, as we had always thought, are comprised of pilin. You change the aromatic amino acid content, the conductivity changes. Oh yeah, so we subsequently now, it's just not published, so I took the data out since we're having recording. You can then also, we have that strain I mentioned before where you express the metalli-reducens pilin that has many more aromatic amino acids. And what we see again is an abundance of the 3 nanometer filaments, but now they're 3 orders of magnitude more conductive than the wild type. So it's clear that those 3 nanometer filaments are comprised of pilin. And I don't know if anyone ever doubted this, if you add a peptide tag to pili, the pilin monomer, such as like a his tag is shown here, and then look at the cells by immunogold labeling or nickel labeling, you see that all the filaments are comprised of pilin. Also, if we express the gene for the Geobacter pilin in E. coli, which of course has no outer surface cytochromes, we get filaments of the same morphology and conductance as we see by Geobacter. So I know there was some pushback on this model, but I do believe that there's just a predominance of evidence that suggests that it's electrically conductive pili that are responsible for this long-range electron transport to iron oxide. It also looks further that these multi-heme C-type cytochrome filaments are very rare They're not significant really or necessary for a lot of types of extracellular electron transfer. This is results from a study just came out, a study organized by Jessica Smith and Xingling Liu. It just shows, this is showing with one of the strains of Geobacter sulfur-reducents, you can delete the gene for OMCS, it has no impact, but now delete the, also insert a poorly conductive pillin, and again, takes away the capacity for iron oxide reduction. Another type of C-type cytochrome that can form filaments in vitro and can be seen in cryo-EM under those conditions. There's no evidence, as was previously published, for the formation of filaments in biofilms of this organism. And when we've looked directly by AFM, also do not see any of these OMCZ filaments. So I think the big question now in this e-Pili or protein nanowire model has been, well, what is the role of some of these other outer surface C-type cytochromes? One guess is that they're helping promote electron transfer from the porin-cytochrome over to the conductive pili, but that is pure speculation and needs to be further evaluated. Okay, so that's one aspect of electroactive iron-reducing microorganisms. I'd like to move on then to look at this other phenomenon that's important in sulfate-reducing and methanogenic environments, known as direct interspecies electron transfer, or DIET for short. And this DIET concept actually came out of the understanding that Geobacter species could directly interact with electrodes. So they can function as what's called an electrogen, producing current by oxidizing organic compounds and now using an electrode rather than iron as the electron acceptor, or with a negatively poised electrode, electrons can flow in the reverse order and support anaerobic respiration like nitrate or fumarate reduction. So this idea of electrotrophs and electrogens led to the concept of whether microbes might be able to function together in consortia as electrotrophs, one being an electrogen, one being an electrotroph, and that turns out to be the case. It's probably most important in methanogenic environments, especially some anaerobic digesters. It's clearly the dominant method is this DIET from between Geobacter species and methanogenic organisms. It's also important, at least in some methanogenic soils and sediments, such as rice paddies. So this DIET process was first discovered by Zara Summers et al. In studies where she combined actually two Geobacter species. So it's a real laboratory artificial consortia, but they were placed under conditions where they had to share electrons in order to grow. And they eventually evolved to do that, forming these large millimeter diameter conductive aggregates, which are connected by a very dense network of the electrically conductive pili. And in these Geobacter systems, you have to have electrically conductive pili. And I think the reason for that, when you look at these fish images, so metalli is green, sulfur-reducents is red, you can see that there's microcolonies of the two different species separated from each other. So in order for electrons to say get from these metalli-reducents to these sulfur-reducents, you've got to have some form of long-range electron transport. However, I really want to emphasize that the e-Pili are not necessary in all cases of DIET. It's certainly possible without that. That was clear in a recently published study by Xingling Zhang, with a newly discovered Methanobacterium species, which can serve as the electron accepting partner for DIET. And here the pili were not required. And I think the reason for that is probably clearest in the scanning electron micrographs. So the Geobacter are the small rods, the Methanobacterium are the large rods, and you can see they're in close physical contact. So there was really no need for very long electron transport, so no requirement for the e-Pili. However, that's not always the case. These are results recently published by Don Holmes, Jinjae Zhao, and others. I'm not sure why it's doing that. And in this case, if you use that strain, it's that Arrow5 strain of metalli-reducents with the poorly conductive pili and try to form a co-culture, it doesn't work. Whereas with wild type Methanosarcina acetivorans, as Amelia nicely pointed out earlier today, can form one of these DIET co-cultures. And the e-Pili were first found in Geobacter, but it's turning out that these similar conductive filaments have independently evolved many times in diverse phylogenetic groups. Many of these microbes, they have the conductive pilin-based filaments, but they lack outer surface cytochromes. So the two are not necessarily coupled together. And here I think is an important example of that. This is Centrophus Acetotrophicus. And this organism is basically the poster child for electron transfer between microbes degrading organic compounds and methanogens, but it was primarily studied for many years due to its ability to produce hydrogen gas from those organic compounds, which the methanogens would then utilize. It was thought that Centrophus didn't have pili, but in fact it does, as can be seen in this transmission electron micrograph. And it turns out that these pili, because they're aromatic, rich in aromatic amino acids, are conductive, a little bit more conductive, in fact, than those of Geobacter sulfreducens. And upon finding that, we asked, well, is Centrophus capable of also serving as a partner, as the electron donating partner for diet? And it can. And the electrically conductive filaments are not found only in bacteria, they're also found in archaea. This was first shown in studies by David Walker on the methanogen Methanosarcina hungadiae. This is an atomic force microscope image. Here's the long archaeolum emanating from the cells. So these archaeolum are homologous to the type 4 pili that are found in bacteria that assemble into conductive filaments. When you look at the archaeolum a little more closely in a conductive scan, they are electrically conductive, and in fact, substantially more conductive than those of the Geobacter pili. And I don't think we really know what the conductive archaeolum do in Methanosarcina. It's difficult to study because it's not yet genetically tractable. But it's a really useful model, the archaeolum is, because the structure of this filament has already been solved by cryo-electron microscopy. And it's really apparent within this structure that there's a core of these aromatic amino acids, which are very likely to be the conduit for the electron transport along the length of the filaments. And this is just another image showing how these aromatic amino acids pack together to facilitate the electron moving through that aromatic core. Not much else is known about archaeolum yet. Sometimes we see archaeolum that appear to be involved in diet. This is from that study recently published by Don Holmes and Jinjae Zhao and others, that co-culture of Methanosarcina, acetivorans, and Geobacter metalli-reducens. And you can see in transmission electron micrographs often that this archaeolum seems to be connecting the two cells. You have to be careful about this. You don't want to just see what you want to see, but you can see those archaeolum there. And it's interesting, if you delete the gene for the archaeolum in acetivorans, then the capacity to form these diet-based co-cultures goes away. And we haven't hadn't had time to measure the conductivity yet, but these archaeolum also have a very high abundance of aromatic amino acids. It suggests they might be conductive, but it's all really speculation at this point. The other important factor in whether or not those archaeolum are conductive in diet is how do these archaea manage to get electrons inside and outside the cell? And this question has really been stymied by some of the most interesting archaea that can get electrons outside the cell, such as the methane oxidizing archaea, are not available in pure culture. They're a little bit difficult to grow and they're not genetically tractable. But we have made a little bit of progress with Methanosarcina acetivorans. And initially, this are studies by Don Holmes, and Toshiyuki and Jinjae Zhao, primarily doing this work. First, looking at how do the electrons get out of the cell, looking at iron reduction or showing reduction of the another extracellular electron acceptor, which are humic acids or humic analog AQDS. And a key feature of this extracellular electron transfer appears to be this multi-heme C-type cytochrome, which we've designated as MmcA. So the archaea don't have those porin-cytochrome systems, as I showed earlier for the gram-negative microbes, but they do have something at least somewhat similar in having this large multi-heme C-type cytochrome on the outer surface. It's important for electron transport out, and it also appears to be important for electron transport into the cell. And I say that because if you delete the gene for that cytochrome, we don't get we're not able to form the diet co-cultures. So by transcriptomic analysis of acetivorans when it's growing with data and based on that gene deletion study, then it's possible to construct a model for how electrons may get into the cell. I don't know if the archaeolum first get the electrons near the cell, but they've got to get across the outer surface. It seems almost certain that it's MmcA, it's the important electrical contact here. And then through the upregulation of these various components during growth on diet versus growth on acetate, it appears that, and Amelia talked about this in detail, I'm not going to go into it, these other components are also involved in electrons getting in. And Amelia covered this real nicely, so I'm going to quickly go through the next couple of slides. It turns out, as she mentioned, the Methanosarcina are not all the same. They fall into two physiological groups. There's type 2 and type 1. And so acetivorans is a type 1, and they have that outer surface multi-heme C-type cytochrome. Methanosarcina lack that cytochrome. So that raises the question, as Amelia brought up, how do electrons get into these type of Methanosarcina? There are a number of putative redox-active outer surface proteins that are upregulated when Methanosarcina barkeri is growing via diet, but nobody's done the functional genetic studies to determine which ones are involved. I think a really big question is for Methanothrix. So Methanothrix are the dominant methanogens involved in diet and anaerobic digesters. They're also very abundant in methane-producing soils. They don't have C-type cytochromes. I think it's going to be really important to figure out how electrons can possibly get into these cells. It's been speculated that maybe the amyloid sheath of these microbes are conductive, and that's a really interesting hypothesis that should be experimentally evaluated. And I think it's just going to be that there's just a wide diversity of mechanisms for electron uptake in the methanogens. They're not all the same, just like all bacteria are not all the same. And for example, this Methanobacterium strain YSL that I mentioned earlier, that can interact with metalloreducens without metalloreducens making pili, probably has yet an even other mechanism for electron uptake. Okay, so I'd like to move on then to this concept, which is also diet, but it's found mainly in sulfate-reducing systems in which methane oxidizing archaea directly transfer electrons or have been proposed to directly transfer electrons to a sulfate-reducing partner. And this anaerobic oxidation of methane is very important in the global cycling of methane. The limiting factor, one limiting factor has, as I mentioned earlier, I'm sure somebody will get this, but it's been difficult to culture these methane oxidizing archaea so far, and certainly no genetic systems for them. But there are some sulfate reducing candidates that might help give us some insight into this. So that's what we decided to look at. And instead of trying to grow them by diet, because those anaerobic methane oxidizing systems grow slowly, we decided to use metallic iron as the electron donor. And the reason that we did this is based on previous studies with Geobacter, where we found that metallic iron can serve as a direct electron donor to support anaerobic respiration. So you can see here these cells of, I believe this is sulfur reducens, growing on this metallic iron. They're directly extracting electrons from the metal. And sulfur reducens, they don't need the pili because they're, as you can see, they're all closely contacting the metal, but they do need some of these outer surface cytochromes I mentioned before, the OMCS and OMCC, in order to support their anaerobic respiration. And this is just a model for how that works. So it's just all speculation right now, but we know OMCS is needed. We know that we think we know the porin-cytochrome system is needed to get the electrons in the cell. And we also know that OMCS, in addition to existing as filaments, actually is very often found just bound to the outer surface. We've known that for some time. So it's likely that OMCS is helping provide more electrical contact, feeding electrons to the porin-cytochrome, and that would be the same idea for OMCC. So the question is, well, what sulfate reducers to work with? And we thought we had some really good candidates. These two sulfate reducers, Desulfovibrio ferrophilus and Desulfopila corrodants, which were previously reported to directly extract electrons from iron metal. It's really important to recognize, though, that direct extraction of electrons from metal was inferred. No genetic studies were done. And I'll show you that actually we ran into a few problems with that. So in the initial studies, we just wanted to confirm, and these are studies by Dan Liang, Trevor Woodward, Don Holmes, Jessica Smith, and others. In the initial studies, we just want to confirm the fact that these two sulfate reducers could use, in this case, pure zero-valent iron as an electron donor for sulfate reduction, and they can. Clearly, loss of sulfate over time when they're grown with zero-valent iron. The issue with zero-valent iron, though, is that it produces hydrogen. In this case, this is the case even under abiotic conditions. Note here on the right, substantial accumulation of hydrogen over time in the standard growth media. So since these organisms can use hydrogen, it's really questionable how they're getting their electrons. The better way to look at direct electron transfer from an iron metal to a microorganism is to use stainless steel, because as studies by Hyung Tang and others demonstrated, this was just published earlier this year, with stainless steel, there's no hydrogen production. Note the difference in scale here. So Dan then did the studies with stainless steel and found that neither Desulfovibrio ferrophilus or corrodants were able to use stainless steel as an electron donor for sulfate reduction. Dan tried with other electron acceptors, and most notably iron, which both, as I'll show you in the next slide, I believe, these sulfate reducers can use iron as an electron acceptor, but with stainless steel as an electron donor, nothing happens even after a couple months. In contrast with either metalloreducans or sulfur reducans, there's direct electron transfer with basically reduction of all the iron that we provided within a week. So the stainless steel definitely can give up electrons to support anaerobic respiration in microbes that can directly withdraw electrons from the stainless steel. So it doesn't look like those sulfate reducers are going to be our model for how electrons get taken up, say, in some of these diet cases. But I think ferrophilus is a really exciting iron oxide reducing microorganisms, because many sulfate reducers can reduce metals, but in cell suspension or under conditions where they really don't grow. And ferrophilus then is one of the few sulfate reducers that actually grows quite well, as shown here, with iron oxide as the electron acceptor. It only incompletely oxidizes lactate to acetate, so kind of like Schumanella in that regard. But I think we're just happy to have any kind of desulfovibrio that's known as iron oxide reducer. So we've been trying to figure out, well, how would electrons get outside the cell of a sulfate reducer? There's no apparent porin-cytochrome genes in ferrophilus, but there are genes for a number of multi-heme C-type cytochromes. And so Dan and Don looked at gene expression of the expression of these cytochrome genes with sulfate as the electron acceptor versus insoluble iron oxide and soluble iron as the electron acceptor. It's a typical strategy used to try to identify genes involved in iron oxide reduction. None of the genes are upregulated during growth on iron oxide. So that doesn't mean they're not important in iron oxide reduction. It's just that from transcriptomics, we don't get that early helpful hint. What was very highly upregulated during growth on iron oxides were the genes for the putative pilin in ferrophilus. You can see this dramatic increase in transcript abundance with iron oxide as the electron acceptor. And what was interesting about that is this putative gene in ferrophilus has a high aromatic amino acid content and small gaps in between the aromatic amino acids along the length, those are the characteristics that predict whether a pillin is likely to yield electrically conductive pili. So this is what the cell looks like in transmission electron microscopy. It has a flagella, but it also has these thinner filaments, as you can see highlighted by the white arrows. Those are pili. When you look at this now with the atomic force microscopy, here again is the thicker flagella, but here's some pili coming out from the cell. You can see it's got a smaller diameter. It's a 4 nanometer diameter pillin. And the flagella is not electrically conductive, as you might expect, but there clearly is a current voltage response by the 4 nanometer filament. When you look in the linear range here, the conductance of these pili is it's about a quarter of that of self-reducing. So it's hard to say. It seems like that might be conductive enough. We need to study this more. We really are interested in studying Desulfovibrio because we've known for a long time that they can be important in iron oxide reduction in marine sediments. Studies way back when by Max Coleman, David White, and others demonstrated, for example, siderite production in coastal marine environments. It was all implicated with Desulfovibrio, but we never could find the right microorganism to study this process. And it's also interesting because it lacks these porin-cytochrome conduits, so it's likely to be a novel mechanism for electron transport outside the cell. Unfortunately, Toshiyuki at UMass has been able to develop genetics for this strain, so we hope to learn more about its electroactivity in the near future. How am I doing on time, John?
I guess, well, we've got a little bit of discussion time at the end, Eric. So another 5 minutes or so would be
5 minutes.
Good.
Perfect. Yeah, whatever you think is right. Yeah. I did want to kind of mention, and I guess kind of looking forward for electron microbiology, anyone that's kind of interested in getting in this field, here again is this chart I showed earlier, the grand diversity of electroactive bacteria and archaea. It's really important to understand that almost none of these microbes have been studied in any real extent. As you can see, we hardly understand a few features of Geobacter or the methanogens. There's all these other microbes that are just super interesting, and I think somebody should be studying them. I think the archaea are especially attractive. I'm hoping somebody can culture out those anaerobic methane oxidizing archaea that have extracellular electron transfer to metals and other organisms. Archaea, I think, are probably going to be a very fruitful area. We've known, for example, that the hyperthermophiles, other than their ability to grow at high temperature, their most highly conserved characteristic is the capacity for extracellular electron transfer. But basically, nobody knows anything about how they carry this out. But it's really going to be important to be able to grow these organisms. I think the idea of, I love metagenomic analysis as a place to start, but the idea of just speculating on what microbes do based on metagenomic analysis, it gives you the just so story, but you've got to be able to capture these microbes in culture and develop the genes, and also learn how to keep them under environmentally relevant conditions. Otherwise, they change if you don't put them in the freezer. And just want to point that out as kind of looking forward to where the field might go. Another way the field is going, it's not really electromicrobiology, but it's using some aspects of electromicrobiology, is the production of sustainably produced or green electrons. And we've been interested primarily in the electrically conductive pili and have moved away from Geobacter to now develop a chassis for E. coli that can produce the electrically conductive pili of Geobacter. And I just wanted to point out since this was an applications session, some of the interesting applications that have been developed in the laboratory of Jun Yao, my colleague in electrical engineering here at UMass. You can just take the films of these wires and lay them down, in this case, between two electrodes of different sizes, and produce electricity just out of the humidity that's naturally present in air. And it's just been amazing what they've done in June's lab. They've developed, again, from thin films of these wires, a neuromorphic memory electronic devices that function like neurons. And the really key aspect of this is with these nanowire memory resistors, they function at biological voltages, which is really key. And then I think a big focus of the research now is that since, as I showed you, we can put different peptide tags on the outer surface of those electrically conductive pili, we can tune their sensitivity for binding various analytes and have been able to make a variety of wearable biomedical sensors with that approach. I do want to point out, I keep mentioning UMass, but I've also started an institute at Northeastern University in Shenyang, China, and we have lots of openings. Oh, we have brand new lab, buildings, equipment, lots of openings. If anyone's interested, please contact me. These openings are at basically all levels. And with that, I'd like to say thank you very much.
Brilliant. Thanks, Derek. That's a really, really excellent overview of some of the underpinning work, but also really exciting to hear about the new updates as well. So thanks so much for sharing those with us. We'll give people a few minutes to add questions into the chat. Maybe I could just start the ball rolling. It's always nice to hear, the message that you've really got to be able to grow these organisms to understand them. I think all the sort of more traditional microbiologists, that's music to their ears. I mean, with all the advances in understanding these sort of more complex interactions, does that help us guide some of the cultivation approaches? Because it just seems that getting organisms out in isolation, is that what's limiting us? Are the ways of getting these symbiotic organisms out using slightly more complex approaches, do you think that's what's holding us back in some areas?
Yeah. I really wish I knew. Otherwise, we could culture them. But I think you're probably right. I think especially for these microbes that are involved in diet, they'll never be recovered by standard enrichment and culture methods, which always select for the most rapidly growing microbes, which are almost always going to be, in that case, a hydrogen-producing microbe. There are ways to selectively enrich for those so that where you favor slow growth, like you might actually see in sediments or in some of these anaerobic digesters. There isn't, the selective pressure is way different than what we put on in enrichment cultures. And there's probably I think you're probably inferring there may be metabolites released by one partner that support the growth of the other. And I think some clever people will figure that out. I came from studies of just studying mud and never culturing a microbe, but I think I kind of learned over time, you've got to be able to culture the microbes or it's just speculation, and it wears thin after a little while.
Yeah, no, absolutely. And I guess one of the big challenges in this field is, when we're working in the lab, often we're using model systems that are doubling in minutes to hours, and I suppose in situ, the doubling times are days to weeks, aren't they? And the processes are really quite different.
Right. Yeah. So patience is required. These diet studies, when you're looking at a month or two for the co-culture to grow, and I think it's difficult projects for graduate students. Our system really isn't suited, at least in the U.S., to these, investing a year in an experiment, and that's often the type of commitment that's required.
Yeah, I think that's a really good point. And we often moan in the UK about everything really, especially at the moment. But the funding rounds. If you get it's difficult to get money, and it's very difficult to get money for long-term studies. And a 1 or 2 year grant's just not enough. 3, you're struggling with these sorts of areas. You really need a big commitment, don't you, to deliver on some of these projects. But,
yeah, and I think the reality of almost all of the things that I mentioned when they were first discovered were all side projects. You just couldn't get funding for them. Yeah. No, excellent. Can I ask one more
question just about the corrosion work? That
Sure.
Sounds really fascinating. So, does that mean that stainless, there's an issue there around corrosion, particularly with the iron reducers? What's the impact on the material if they're if they are getting electrons out of the metal surface.
Yeah, so I mean, it is a metal corrosion, and then the question is, just how fast it goes. So I'm very interested in corrosion now. I think it's an important area of microbiology that's been studied for well over 100 years, but not like with modern techniques. So I think it's an economically important project or area of research that could warrant some further study. So it even stainless steel corrodes. So almost all other steel corrodes. And my understanding on the applied end from industry is just guesstimating how fast it's going to corrode. So that there aren't really great prevention strategies in many instances.
Yeah, no, absolutely. One last question, if I may, as well, Derek. I think you answered it in the slide that you said was some speculation, but I think you've shown very elegantly that those pili are obviously crucially important. But those cells are packed full of heme groups, aren't they? Which don't seem to be so important in ion reduction. Is that the same argument as you would give for the outer membrane cytochromes? They're there just to help get the electrons to those pili rather than to the metal directly.
Yeah, so I think it depends. So I mean, in some cases, with the soluble extracellular electron acceptor, and I know you know all this, you don't need a pili because there's enough electrical contact. But I think in some instances, like I showed some of those instances of diet where the cells aren't in close contact, I think you need something beyond an electrical conductive outer surface. So the cytochromes can do that, but for long range reach, you really need something more.
Thank you. So there's a question from Deanna Benning that's just come in. Thanks, Derek. Can you maybe comment on how and if such nanowires would survive in a much more complex natural system compared to a lab culture, and if their role would change in nature?
Oh, yes, that's a great question. I think often comes up. I think the question is, well, if those nanowires are protein, wouldn't some other microbe just come and gobble them up and they'd be... the pili are extremely resistant to proteases and other attack mechanisms that microbes might have, even in the laboratory. I think one of the problems or difficulties in studying these filaments is they're very hard to denature. You have to, they don't even denature under boiling SDS, conditions you would use normally for protein separation on gels. So I don't think it's a problem in nature because they're so resistant to degradation.
Thanks, Derek. Okay. I can't see any more coming in by chat. Lots and lots of messages saying thanks very much for such a fantastic talk, Derek. So a fantastic talk to end the proceedings on.